From the National Institute of Justice's Expert Systems Testbed Project Series, the following presentation is the technical capabilities, concordance study, and examples. The NIJ Expert System Testbed Project, commonly referred to as the NEST project, was launched as a technical assistance project to the forensic DNA community in May of 2005. The goals of the project are to evaluate the commercially available expert systems designed for the technical review of single-source data produced by forensic DNA laboratories, and to report the results to the forensic DNA community. This podcast represents part one of the presentation, introduction, and detailed information on each expert system. Your presenter is Rhonda Roby, a technical consultant for the NIJ.
The NEST project was focused on evaluating the ability of commercially available expert systems as a mechanism for rapid, accurate technical review of convicted offender single-source DNA samples for the eventual upload into the National DNA Index System, or INDES. The goal of the NEST project was to evaluate, not validate, each of the expert systems. Moreover, the NEST project team that conducted this study comprised of not only leaders in the field of forensic sciences, but also users of the different expert systems. This presentation is designed to be informative and non-biased. It is designed to provide the information that the NEST project team has summarized in its evaluation of these expert systems. The primary purpose of the study is to provide the forensic community with an objective perspective on available technology and expert system software for the analysis of single-source convicted offender samples for accurate review. One additional purpose of this study is to educate the managers and scientists in the forensic community and to provide sufficient information so each one is a more informed consumer of the software systems. We believe this extensive study will be of value to those choosing an expert system for their laboratory. The three expert systems that underwent a rigorous independent evaluation of single-source samples for the NEST project are GMap ID version 3.2, FSI Cube Expert System Software, and Trula System 2. The mathematical and statistical programs used to process the data for the different expert systems can be quite simple to complex. Whereas each algorithm for the different expert system programs has not been independently studied, it is clear from the evaluations performed that they do work properly. These expert systems perform functions such as processing the raw image files, removing background noise, applying matrices for die-to-die separation, detecting of peaks and noise, and quantitatively computing peak heights and area and performing calculations with these measurements. The intent of this presentation is to share the different rule firings from the three expert systems. However, you must recognize that there are a number of important features in each expert system that we will not be able to cover in this presentation. Therefore, I am going to discuss a few of these important features at this time. First, I am going to provide a general overview of GMap ID. When GMap ID processes the raw data files, it sizes the peaks, separates the colors, applies complex mathematical algorithms to quantitate the peaks, identifies the allelic ladders, and makes allele calls. The analysis of the size standard in GMap ID is simple. A size quality flag is used to confirm the performance of the size standard. Depending upon the manner in which the file folders are organized, the software either uses the allelic ladder within a set of injections or compiles a composite ladder from all the ladders in a project to assign allelic values to sample peaks. When the user clicks on GMap ID to process the samples, it first processes all of the defined allelic ladder injections, and then it processes all of the samples in a user-defined order. Many features in GMap ID are intuitive. The software has process component-based quality values, or PQVs, for ease in evaluating each sample. The PQVs are the software's expert systems rule firings and are based on predefined specifications. The overall genotype quality is measured as a result of the software performing a calculation based on the individual PQVs for each locus. The PQV results are graphically represented as a green square, a yellow triangle, and a red circle representing pass, check, and low quality, respectively. To use GMap ID as an effective expert system, all loci passing the overall genotype quality are not reviewed. The analyst can quickly sort and identify the check or low quality calls with the push of a button that bring all flag loci to the top of the list, or the analyst can make individual tables to sort identified lower quality samples. The analyst has the option to accept, edit, or reject the allele calls. Another important feature of GMap ID is its control concordance PQV. The control concordance PQV confirms the genotype of a single referenced positive control, or it can be used to compare results from different analysts. GMap ID, like the other expert systems, also allows for the monitoring of the negative controls. GMap ID can produce a CMF version 1.0 and version 3.0 format files for easy import into CODIS. This feature is completely functional and available in its current version. Now I'm going to turn our attention to FSI Cube Software. FSI Cube Expert System Software is a suite of software programs known as iStress, iStream, and Integrity. iStress is the program that analyzes peak information and assigns allelic values. iStress is dependent on the data generated by other software programs, such as GMap ID. When validating FSI Cubed with another software program, such as GMap ID, both programs are used as a system and must be approved as a package. DNA profiles are produced in iStress by applying rules and filters to the peak information generated by and exported out of the supporting software. This presentation is focusing solely on the iStress feature. Since the purpose of this presentation is to evaluate single-source samples, iStream, a mixture deconvolution feature, is not a part of this evaluation. iStream produces a best fit analysis of two-person mixed DNA profiles. Integrity produces a report to identify potential contamination on a plate or batch of samples run together. For the evaluation of FSI Cubed in Phase 1, tables containing peak information generated with GMap ID were created, exported, and subsequently imported into FSI Cubed. FSI Cubed builds from the peak detection and sizing algorithms within the Applied Biosystem software. iStress uses the base pair size, peak height, and peak area information generated with other software to perform its calculations. The evaluation of the size standard is performed via other software prior to the generation and export of the raw files rather than post-import into FSI Cubed. GMap ID is not used as an expert system or as an allele calling program when used as support software for FSI Cubed raw data files. The steps for reviewing the size standards for FSI Cubed are the same as when reviewing the size standard in GMap ID. Therefore, the analyst must have some level of proficiency with GMap ID. Specifically designed panels, bin sets, and analysis parameters recommended by Promega are used in supporting software such as GMap ID. These panels and bin sets are designed to allow for the export of as much peak information as possible. FSI Cube builds from data generated with either GMap ID or other supporting software. The data are imported in tabular format and are used for downstream analysis in FSI Cubed. This process requires the manual transfer of the data tables generated with the Applied Biosystem software programs into the FSI Cubed environment. Once the data are imported into the FSI Cube software, the application runtime is minimal. When using GMap ID in conjunction with FSI Cube software, no samples are identified as ladders or controls. The tables are exported as a text file. The software automatically recognizes the ladders and controls and makes allele calls accordingly. Any software program that can export base pair size, peak height, and peak area in a text file can be used with FSI Cubed. FSI Cubed evaluates each ladder and either passes or fails the individual allelic ladder. An allelic ladder will fail if it does not meet the minimum criteria, such as number of alleles or minimum peak height. All passing ladders in a batch are used to create a consensus ladder. The consensus ladder is then used to make the allele calls for that batch. After the ladders have been evaluated by the software, clicking on an icon initiates the allele calls and rules are applied to the sample data. Processing of the samples is efficient. One useful feature of FSI Cubed is its ability to check the concordance of different positive controls. Many laboratories use the amplification kit, positive control, and an internal extraction control. With FSI Cubed, the software administrator can identify multiple controls that can then be checked automatically with the software. FSI Cubed also allows for the monitoring of the negative control. The rules used in the evaluation of samples for FSI Cubed are optimized and defined by the laboratory. The laboratory system administrator has full control and access to define the multiple rule settings available in FSI Cubed. The rules available for definition can either be set for visualization or not. If they are activated, the triggered rules are displayed during review. The software displays and organizes the samples, triggering a rule one after the other. Samples passing the set thresholds are skipped as the analyst conducts review of a batch of data. Once a rule is displayed, the analyst has the option to accept, edit, or reject the allele calls based on laboratory policy. The values applied to the rule settings are meant to flag or warn the user when a particular criterion or rule is not met. These criteria can be set to the interpretation guidelines of the laboratory. The rules can be default settings or individualized for particular runs, chemistry sets, or samples. Not all rules are intuitive, however, it is easy to find the definition of each one in the materials provided by the manufacturer. Integrity is a feature within FSI Cube software that evaluates all samples in a batch for well-to-well contamination. Integrity compares the alleles in a sample to all other samples in a batch. The user can define the level of allele match stringency in the software. Lowering the stringency decreases the number of required allele matches and increases the number of samples that may match. Raising the stringency increases the required number of allele matches, which makes the well-to-well match much more specific. A summary of the matches made is automatically generated and saved into a file on the hard drive. The integrity program is easy to use, and the display is quite easy to view as a plate map. The different match components would be used to evaluate different situations, such as plate splashover, general contamination from an analyst, duplicate samples, and so forth. We are now going to discuss Trula System 2. Trula System 2 creates a batch or gel image from the imported raw data files and sizes the peaks, separates the colors, applies complex mathematical algorithms to quantitate the peaks, identifies the allelic ladders, compares the ladder peaks to the data peaks, and assigns allele calls. Trula System 2 processes raw data files generated during electrophoresis. For example, if Applied Biosystems capillary instrumentation is used for electrophoresis, the raw.fsa files stored in data collection software are directly imported into and used by Trula System 2. The Trula System 2 software package does not rely on the output data as FSI Cube does. The raw data files must be organized into files known as data disks prior to being run through the software system. The user has the option of setting up data disks that contain single batches of data, or to maximize efficiency, the user can organize multiple batches of data into a single data disk. Organizing multiple batches of data within a single data disk reduces the time and effort required for manual transfer and setup of multiple individual batch files. Analysis is performed on all data included in a data disk. Depending on the number of samples analyzed, this process can take several hours. The program can accept data from Macintosh, Windows, or Unix datasets. Data analysis is a multi-step process, the first being the organization of the data into the data disk. When analysis is initiated, the expert system performs signal analysis and allele assignment. Once the review is complete, a number of output files are generated. Trula System 2 quantitates data by applying complex statistical and computational analysis programs to each phenotype. The system can compare the observed results against the expected results. Large deviations between expected and observed data enable the software to flag potentially problematic data for human review. The analysis of the size standards in Trula System 2 is simple. The image call module of Trula System 2 assigns the values to the size standard while the cap view interface is used by the analyst for review of the size standard. Evaluating the size standard in cap view is conducted with a batch view. For example, all size standards associated with the samples in a batch are presented in a single view. The software translates the size standards from a capillary electrophoresis run and displays a gel image of the size standards. The presentation of the size standard information in this image is an excellent tool designed by cybergenetics that allows the analyst to study each sample independently as well as all samples together. This gel image view provides the analyst information with respect to the size standard associated with each sample, as well as quality assurance information on the entire run. Cybergenetics highlights that the gel image view be reviewed to look for quality assurance issues, for example, migration differences that could be attributed to temperature fluctuations. The control check module of Trula System 2 is used to perform a quality assurance check on a batch of data, including the evaluation of the allelic ladders and controls. A number of options are available to the analyst to review the software's tracking of the allelic ladders. Ladders within a batch and across a batch can be overlaid and the migration differences visualized. This function may also be applied to the size standards. The user can choose to use the closest passing ladder to a sample in a batch to make the allele calls or create a consensus ladder. Within the control check module is the ladder failures interface and the positive control check interface. The software automatically identifies and rejects poor quality loci within an allelic ladder and evaluates the positive controls. At this step, the software searches all of the data within a run. Any sample appearing to be a control, but not identified as such, is brought to the analyst's attention. There is no limitation in Trula System 2 on the number of controls that can be defined. Auto results is a crucial component of Trula System 2. Auto results ensures integrity of data for the CODIS CMF file. All accepted data is processed through auto results to check for incomplete samples, concordance at common loci, and concordance at all loci for samples that have been run multiple times within a run or between runs. Auto results will generate 3 output files: an incomplete file, an inconsistent file, and a CODIS CMF file. The incomplete file lists all the samples that are incomplete. These samples do not get exported into the CODIS CMF file. The inconsistent file lists all the samples that are inconsistent at a locus. Trula System 2 does not include the incomplete and inconsistent files for the CODIS CMF output file, thus ensuring the integrity of the data. The Allele Call module of Trula System 2 applies different rules to all identified peaks and assigns a quality score for each locus. These quality checks enable a user to focus on the samples that have triggered rule firings as a result of having values outside the defined thresholds. The software displays the loci with the lowest quality values first. Presenting the low quality values first focuses the analyst's attention on those samples requiring human review. The analyst has the option to accept, edit, or reject the reviewed allele call in Trula System 2.
This has been an introduction to the NEST project and a high-level overview of each of the expert systems that we will be evaluating in this presentation. In the next podcast, Rhonda will deliver an overview of the concordance study, as well as a visual introduction to positive control and allelic ladder. The content of this podcast was funded by the National Institute of Justice, which is part of the Office of Justice programs within the U.S. Department of Justice. The National Institute of Justice, or NIJ, is the research, development, and evaluation arm of the U.S. Department of Justice. NIJ has numerous programs that support state and local crime laboratories across the country.